if we dig deeper, we see that the brain is not only an electrical machine, but also a structure based on chemical reactions. Synapses play an enormous role in this structure. They are contacts, or in other words, connections between nerve cells. As a rule, a neuron's axon reaches the next cell. This may be another nerve cell, a muscle cell, a glandular cell, and so on. The neuron's axon forms a contact with the cell it reaches. This contact is called a synapse. As long as information remains inside a neuron, it is transmitted in electrical form, as impulses. But when it is time to move on to the next cell, the transmission occurs in chemical form via special substances called neurotransmitters. Put simply, neurotransmitters are like mail couriers that carry conditional documents with information from one cell to another. In other words, they are intermediary molecules. What we get is an alternation: electricity inside the neurons, chemistry between neurons, then electricity again, and chemistry again. This constant switching between modes of information transmission is a fundamental principle of how the brain works. It is precisely at the chemical level that it is much easier for us to influence the functioning of the nervous system. If we know which substances are released in synapses, and science already knows this quite well, we can synthesize and introduce into the body molecules that are similar to them, either to enhance their action or, on the contrary, to interfere with their function. In this way, we exert a serious influence on brain functions. We alter the balance between excitation and inhibition, affect memory, emotions, and behavior. The vast majority of such substances, medications, poisons, or narcotic drugs, resemble one of the brain's primary neurotransmitters. Synapses are extremely important for the brain's operation. Here is roughly what a synapse looks like. Inside the axon terminals are membrane-bound vesicles. They contain neurotransmitters. The logic of synaptic operation is as follows. First, an electrical impulse, an action potential, runs along the membrane of the nerve cell. This impulse acts like a signal saying, "Send the courier," and it triggers the movement of vesicles filled with neurotransmitter toward the next cell. The information is on its way. The vesicles reach the axon membrane, rupture, and release the neurotransmitter into the narrow gap between the axon and the neighboring cell. This gap is called the synaptic cleft. The neurotransmitter then influences the adjacent cell. How exactly? Well, it essentially rings the doorbell like any well behaved courier. On the membrane of the receiving cell sit special proteins. They function as buttons, and our courier, the neurotransmitter, is the finger that presses them. After the doorbell is pressed, a signal saying "send the message" is triggered inside the target cell. As a result, an impulse, an action potential, arises on its membrane, and the information runs further along. The information has been received and passed on. There are also opposite situations when pressing the button inhibits the next cell, and for a while it stops transmitting signals. This, too, is extremely important. In the human nervous system, two principles constantly coexist and compete with each other. One is to transmit information, the other is to avoid transmitting unnecessary information. Both are critically important. This is why some mechanisms ensure the transmission of an impulse to the next cell, while others block it. With this in mind, neurotransmitters released in specific synapses are divided by their effects into two large groups: excitatory and inhibitory. Excitatory neurotransmitters are those that make the next cell work, generate impulses, and transmit important information. Inhibitory ones are those that prevent the transmission of excessive information. Using our courier analogy, they block advertisements and spam. The most important neurotransmitters are glutamic acid and gamma aminobutyric acid, abbreviated as GABA. Glutamic acid is probably familiar to you as a flavor additive. The very glutamate that enhances the taste of almost everything. In the brain, it functions as the most important excitatory neurotransmitter. At least 40% of all nerve cells use glutamic acid as their courier. Thanks to the release of this substance, sensory signals are transmitted, memory functions, centers of thinking and decision making operate. Motor program until they reach the muscles also depends on the release of glutamate. Gamma aminobutyric acid, abbreviated as GABA, is used as an inhibitory neurotransmitter that blocks the transmission of excessive information in at least one third of all neurons. That is how important it is. This substance prevents unnecessary signals from being transmitted and restrains informational noise in the nervous system, noise that interferes with processing truly important data. This task is no less significant than signal transmission itself. Just imagine being shown two movies at the same time in the cinema with a commercial thrown in on top of that. You would go crazy. It turns out that the brain works well not when many neurons are excited, but when only the right neurons are active. Perfectly, their number should be small. There is a popular question. People love to ask it. At any given moment, only about 10% of neurons are actively functioning in the brain. How can we make more of them work? The answer is it can be done, but you don't need it. If too many nerve cells start generating impulses simultaneously, chaos arises in the courier system, and the brain becomes overexcited. It may even result in an epileptic seizure. Such an accident is clearly unnecessary. A well functioning brain is not the one that has activated all its cells, but the one that has managed to engage the right ones, specialized for the current problem. Suppressing noisy neurons is a crucial task, and gamma aminobutyric acid handles it brilliantly. We have now briefly become acquainted with the two main players in the brain activity: excitation and inhibition. Going forward, we will be more interested in second level neurotransmitters responsible for emotions, motivation, and needs. They mainly generate positive emotional experiences. At moments when a person, from the standpoint of our biology, manages to do something beneficial, they are, so to speak, messengers bearing good news. For example, you ate a tasty soup, learned a new way to do cross stitch, especially if it fits your interests, or successfully escaped from an angry neighbor's cat. At such moments, neurotransmitters are released in our brain: dopamine, norepinephrine, endorphin. These couriers bring good news and lift your mood. In fact, the list goes on. There are about a dozen neurotransmitters associated with the satisfaction of needs, and positive emotions, and we will gradually get to know them. A neuron is sometimes compared to a computer chip, and a very complex one at that. A single neural cell has, on average, 3,000 to 5,000 synapses, that is, connections with other cells. Each neuron simultaneously receives information through thousands of channels. Some of them are excitatory, others inhibitory. The neuron must decide whether to pass the signal on or block it by comparing their activity. Individual neuronal chips are assembled into computational centers responsible for breathing, responses to sound, short term memory, and many other processes. An incredibly complex network. Even an international courier service like DHL can compete with it. This analogy is already too modest. The brain can be compared to a vast computing center in which thousands of individual processing units interact with one another in very complex ways. So, how many neurons are there in our brain overall? The number usually cited ranges from 85 billion to 90 billion. Try to imagine that nearly 100 billion. That is far more than the number of people on planet Earth. Just picture 90 billion subscribers, each simultaneously communicating with 5,000 of others. The complexity of information flows inside our head is comparable perhaps to the entire internet, and even that comparison is a stretch. Science and modern technology have only just begun to understand the brain and neural networks. Large scale processes are relatively easy to observe, but the fine details of information transmission still require enormous effort. At the same time, brain cells are very small. The most common size of a neuron's cell body is from 300ths of a millimeter to 500ths of a millimeter. The average weight of the human brain is about 1,300 grams. In men, it is roughly 100 grams heavier than in women. When this was first discovered, the male half of humanity became terribly proud. Later, however, it turned out that neurons are surrounded by glial cells. These are the support staff. They protect neurons from mechanical shocks, monitor the chemical environment, provides electrical insulation, and perform many other functions. It is mainly glial cells that account for the greater weight of the male brain. The number of neurons in men and women is approximately the same, 85 billion to 90 billion. There is also a correlation between brain mass and total body mass, and this parameter is much more stable than the overall weight of the nervous system. As a result, the male brain is better packed and more resistant to blows. Historically, this makes sense. Men led harsher lifestyle while hunting mammoths and asserting themselves as tribal leaders. The female brain, in this sense, is more delicate and sensitive. It's not designed for rough handling. Most neuron cell bodies are located in the brain and spinal cord. but more than 100 so-called small brains, known as ganglia, are scattered throughout the body. These ganglia also contain neurons. Some handle sensory signals, while others work with internal organs. They are small control centers that make minor decisions without consulting the main management, the central nervous system, although they ultimately remain subordinate to it. From the ganglia, as well as from the brain and spinal cord, nerve processes extend. They bundle together into nerves that work with muscles and organs. Within nerves, opposing information flows often coexist. Signals from sensory organs travel to the brain, while commands are sent to muscles and internal organs. To control peripheral devices effectively, it is essential to know how they are functioning and what is happening at the boundary between the body and the external environment. When an axon heads toward the next cell, that cell may be a neuron, a muscle cell, a heart cell, or an intestinal cell. So synapses exist not only inside the brain. From the perspective of cytologists and scientists who study the internal structure of cells, the neuron is built in a fairly standard way. Despite its extravagant appearance due to numerous processes, internally it has an ordinary structure: a nucleus, mitochondria, ribosomes. Its metabolism differs little from that of typical cells. The key difference is that neurons consume a great deal of energy. By this measure, the brain ranks first in the entire body. It requires more glucose and oxygen per gram of weight than any other organ. That is why, when supply problems arise, the brain is damaged first. The kidneys rank second in energy consumption, and the third is the heart. But the brain remains the undisputed leader in metabolic intensity.